Welcome to the first of the vignettes. It's about biological puzzles and probabilistic thinking. So, what I want to do in the next couple of vignettes is try to really drill down into the way we use probability distributions to clarify our thinking about a whole myriad of different questions. I like this particular figure that I'm showing you right now because it gives you four of the key distributions that we're going to focus on. And in each case, there's a little biological story. And then there's an example of the kind of data that comes out of examining that particular question. And it tells us what the null hypothesis is from the standpoint of the underlying distributions. So on the left, the first one shows the waiting times between steps for a molecular motor. Now, this is clearly something that it looks like it's got some regularity. And so the question is, what's the nature of that distribution? And can we understand mechanistically why it takes the shape that it does? The next one, the middle one, it says we eat some bacteria. If each of our cells is equally likely to be infected, our null hypothesis for bacteria per infected cell is the Poisson distribution. And so I'm going to actually use the case of mRNA distributions or mRNA counts as a parallel way of looking into the Poisson distribution. The next one is the binomial distribution, and that has to do with the distribution of alleles, for example, of eye color in flies. And this is something I'll come back to later in the course. And then finally, on the right-hand side is something we're going to dig into next week, which has to do with diffusion. So with all that as the background, the way that I want to couch this particular vignette is through 3 or 4 distinct biological puzzles. And then I want to just give you a flavor for the way that probabilistic thinking insinuates itself into the way that we would consider those problems. So the first thing that I want to mention is that much of the world's photosynthesis takes place in the oceans. And that photosynthesis is done by single-celled organisms. They're known as cyanobacteria. And they carry out something between 10 and 30% of the photosynthesis. I've seen various numbers before. And they have these beautiful structures. So the point is that one of these cyanobacteria has a length typical for a bacterium of, let's say, 1,000 nanometers or a micron. And inside, it's filled with these organelles that you see on the right, which are known as carboxysomes. And these carboxysomes are compartments that hold the enzyme complex known as rubisco, which is responsible for carbon fixation. Later in the term, we'll actually talk about why compartmentalization might be advantageous for the purposes of ramping up the rate per enzyme of an enzymatic reaction. You can see here from these cryo-electron microscopy images that the number of rubiscos per carboxysome is on the order of something like 250. So if we were to look at the cyanobacteria under a fluorescence microscope, in the case in which these carboxysomes had been labeled, then what we could do is we could count the number of carboxysomes in each one of these cyanobacteria, and then we can ask the simple question, what is the nature of that distribution? First, what would we expect? And we're going to use this notion of null model repeatedly throughout the term. And our null model, at least mine, would have been that there's random partitioning. What do I mean by that? I mean that every time one of these carboxysomes is going to be passed to the daughter cells, it's going to flip a coin and either go right or left, sort of with equal probability. And so what that would lead to is what's known as a binomial distribution of counts. And that's shown in the bottom left in brown. In other words, if it truly was a coin flipping situation, then you would get the brown distribution. But what's actually observed is the green distribution. So this tells us that our null model is actually flawed. And that's really cool because in a way, this is going to be an example of a huge theme for this whole course, which is the nature of defiance in the history of life. And what I mean by that is that lots of things that biological organisms do are in defiance of the natural sort of equilibrium or maximum entropy realization of that particular system. And what's especially cool in this case is that by breaking the apparatus that's responsible for partitioning and making sure that both sides get equal numbers, then it does indeed become a binomial distribution. A very beautiful one, really one of these experiments to change your life for. Okay, so the next example so that was the binomial distribution. The biological puzzle was partitioning, as I mentioned, and the insight came by looking at it through the prism of the binomial distribution. And this is just to foreshadow. In other words, I'm not expecting you to know what the binomial distribution means in detail at this point. It's something we're going to work out in great detail in the next vignette. For now, it's just to whet your appetite. So the next example has to do with the opening of ion channels. So in a neuromuscular junction, as you see on the left, information from our brain travels to our muscles. Neurotransmitters are released at the gap between the neuron and the muscle, and neurotransmitters have to diffuse across that gap, and then they bind to ion channels, and those ion channels will then open up, leading to a transient change in the permeability of the membrane. This is just to remind you that there's all sorts of different kinds of ion channels. And when I say that, what I mean is that these ion channels are gated by different driving forces. On the left, you see that voltage can be an indicator to a channel that it needs to open. The two middle examples are cases where a ligand binds to the channel. And by virtue of that binding, it opens or closes the channel. And then on the right is a schematic of a mechanosensitive channel where there's some sort of coupling to tension, which will open or close the channel. Now, how do we know about what goes on in these channels? This is really the first of the single molecule experiments. You take a patch of membrane, you excise it from the membrane with a pipette, and then as shown on the right, you insert it into some solution. The pipette has an electrode in it, and then what we do is we measure the amount of current passing through the channel as a function of time. And what you get is one of these telegraph-like signals shown on the bottom right. And this is the essence of the challenge that we want to face. And that is, you can see that there's a distribution of times. In other words, I have to wait a certain time before the channel opens, and then while it's open, I wait a certain amount of time till it closes again, and vice versa. It keeps on doing this. And the question I ask is, what can I say if I measure this a bunch of times over and over and over again? I can get a distribution. I can plot a histogram of how long I wait between openings or between closings. The question is there some sort of way that by pure thought I can understand what the nature of that distribution is or should be? And this will lead us to consideration of the so-called exponential waiting time distribution. So that I will work out in several vignettes from now. So that's a second example. It's an exponential distribution. The next example that I'm going to be interested in is of mRNA in cells. So if you imagine I take a mammalian cell, a yeast cell, a bacterium, and I ask the question, in a population, I look at them one by one under the microscope, and I fluorescently have labeled the mRNAs for some species of interest, then I would say, how many do I get in cell number 1? How many do I get in cell number 2? And so on and so forth. And once again, I can plot a histogram of the counts of mRNAs. So I might find some cells have 0, some cells have 1, some cells have 2, and so on. And that will have some characteristic shape. And my question is, can I reason out what that distribution should be like? Now, the underlying model, the null model that I would have of how this works is of what is called a constitutive promoter. The idea being that the promoter is basically a factory. It's a factory for RNA. It's just basically cranking out mRNA at a rate r. The RNA is in turn degraded at a rate constant gamma. And so what we would do is we'd write down some governing equations for this, and we would actually be able to work it out. Now, in terms of the measurement, I just told you a minute ago how this goes. You might have some scheme for labeling the RNAs, such as is shown on the right, which would be FISH. You basically have probes that are fluorescently labeled. They hybridize with the mRNA, and that allows me to count to 1. And then I figure out how many such mRNAs are there per cell, and I make a distribution like you see on the right. Same thing can be done on the right-hand side using techniques like MS2, which we'll come back to later. But the upshot is this, that one can count the mRNAs on a single cell level, and one can ask, what's the nature of the resulting distribution? In this case, on the left, we see what will turn out to be the Poisson distribution. Not universal for mRNA distributions, but certainly in some cases it turns out to be Poissonian. The final of the key distributions that we'll be thinking about is the so-called Gaussian distribution, and I just wanted to give you a characteristic example of the kind of place where this will show up, is in looking at a fluorescently labeled motor. This is a motor that actually moves randomly on a microtubule, MCAC, you see at the top. And what we can do is we can keep track of the position of these motors as a function of time. And if I do it for a bunch of motors and I plot their distance from where they started as a function of time, it will be a Gaussian shape. And the longer I wait, the wider the Gaussian will be. So that's something that we will work out in the context of the diffusion equation in a later vignette. So we will also be able to think about this in terms of the binomial distribution. That's kind of fun because we'll see that the Gaussian can be thought of as the large n limit, the large number of coin flips limit of the binomial distribution. And as I said, what will come out of that is a Gaussian distribution. So what I've told you in this brief vignette is really nothing more than to try essentially an introduction to 4 of the key distributions that we'll be spending a lot of time focusing on in the next several vignettes, and then that will rear their heads repeatedly throughout the remainder of the course. And just to remind you, so the top left, the first one I talked about was the binomial distribution. The second one I talked about was the exponential distribution. The third was the Poisson. And the fourth was the Gaussian distribution. And what I would say is that these 4 distributions give us a lot of horsepower. 80% of the time, I would think that distributions arise in our thinking about the nature of the world around us. It's going to be one of these 4 distributions that will allow us to peer into mechanism and understand the nature of the signal that's actually lurking in the noise. And that's going to be, in a way, a mantra of this part of the course, is that there is signal in the noise. We've said earlier, quantitative data demands quantitative models, and now I'm telling you something even sort of more refined, which is that one of the kinds of quantitative data is not the mean, but in fact the whole distribution. And by examining the whole distribution, we can learn a lot about what's going on with our system. So with that, I'm going to wrap up this particular vignette, and I look forward to the next.